All right, so now we've taken the first step in the central dogma from DNA to messenger RNA. Now the question is, what do we do with that messenger RNA? We now have to take this information, AAG, UAU, and so forth, and translate that into amino acids. What does AAG mean in amino acids? And what does UAU mean in amino acids? So this is the translation step. We go from nucleic acid to protein. So how do we do this? Well, it's translation. Just like any other translator, to do this, you need a dictionary, a translation dictionary, which shows you what words are in one language, what they mean in the other language. So the question then is this: what does the word AAG in nucleic acids mean in the language of proteins? So just like any other, we need a dictionary. And there is one. If you look in your textbook, you'll see a dictionary which looks much like this. And this dictionary gives you every possible word in the nucleic acid language, because again, the words are always 3 letters long. This is every possible word. Now, we don't memorize this chart, unless you unfortunately work with this so much that you actually do memorize it by accident. The purpose for this is not to memorize it, but to use it, be able to use it. So here's how this is used. The way it's organized is this. We look at the first letter of the codon. We find here these 4 possibilities, UCAG, not T. Remember, we're looking at the messenger RNA right now. So notice that everything in this row has a C. Everything starts with the first letter is C. Then the second letter is up here. So this is again UCAG. And notice everything in this second column has a second letter of C. And then finally, the third letter in the codon is given to you here, UCAG. So the question then is, how do we translate AAG? What does that actually translate into? Well, first codon is A, second codon is A, third codon is G. So we go here, A, G, there it is, and we would then get lysine. So AAG translates to the amino acid lysine, and that is the amino acid in this part. Now remember, this is the 3rd exon of the beta-globin gene, so this is not the start of it, this is towards the end of it, the 3rd of 3 exons. Now what I would like you to do is to take these last remaining codons and use this chart on your own to translate the rest on your own, and then stop the lecture, do that, and then come back and we'll talk about it. All right, so we have now that the AAG is lysine, and if you've done the rest, you've discovered that UAU becomes tyrosine, and CAC becomes histidine, and UAA doesn't become anything. It's a stop codon. So let's go back to that and take a look at this. The stop codon is UAA here, and notice there's 2 other stop codons, UAG and UGA. There's no amino acid associated with any of those 3. And that's for a reason which I'll explain when I get into the mechanism of this. But these are the stops. These are all the 3 possible sequences that will stop a coding region. And every coding region stops with one of these 3 sequences. Now, there's another special one here. Notice this, AUG. And that is the start sequence. Notice when we began, I said the AUG sequence was always a start of the coding region. So the green AUG here is the start of a coding region. The red, 3 of these reds are the stop coding region, and everything else in between is the amino acid that comes in this way. Now, this AUG, notice, says methionine and start codon. So the question is, which is it? Is it a methionine or is it a start codon? And the answer is yes. It is both. It's always a methionine, but it's always a start. And what that means is that the proteins always start with methionine when they're first built. They're always built first with methionine. Now, if you go back and you look at the actual sequence of the beta-globin gene, you'll notice that the very first amino acid is not methionine. And the reason for that is during the secondary processing, after the protein is built, that methionine is cut off. But still, every protein that's built is built starting with methionine, except for a few oddball prokaryotes. There are some that don't have a start codon that starts with methionine, but all eukaryotes do, and most of the prokaryotes actually start with methionine. All right, so what you're looking at here then is the last portion, the very last 3 amino acids and the very last 4 codons of the 3rd exon of the beta-globin gene. This is how it stops. It goes lysine, tyrosine, histidine, and then it hits that stop codon, and that's where the protein stops being built. So the question then is, how does this work? How does this actually occur? So we've gone through then conceptually the transcription step and the translation step, And now what we want to do is to get into mechanisms. And what we're going to do in this lecture is to look at this. We're going to first look at transcription. Then in the next lecture, we're going to look at translation. We're going to look at these mechanistically. What precisely is happening during these processes molecularly? What's happening at the molecular level? That's our goal right now, to work through those. With transcription. Remember, in transcription, our goal is to make a copy of the gene. And here is an example of the DNA. There's major groove, minor groove, all that stuff that you already know. And here's our gene. Suppose our gene is in this region, and right here we have this green box that's labeled the initiation site. The initiation site is in the promoter, and for beta globin, that initiation site is the TATA box, which again, remember, is CATAA in the human beta globin gene. In normal genes, it's TATA, which is why it's called TATA and not CATA. This is not the start codon. This is critical. This right there, that initiation site, is not the start codon. It's the part of the promoter that is going to attract the copy machine. So what happens is this. What you've got to do is first, like we do with DNA replication, which is very similar to this process, what we have to do is to denature the DNA. We have to separate the two DNA strands. Now remember how that happened in DNA replication. And in fact, this is unfortunate. It's going to be so similar to DNA replication that this process is going to be easily confused with the basic mechanism of DNA replication, because it's trying to do the same thing. It's making a copy of the nucleic acid. And in fact, it evolved from the same set of genes. So again, mechanistically, it's very similar. So what I'd like you to do is to think through this and compare it and contrast it to DNA replication. Compare and contrast transcription to DNA replication. That's a good way to keep straight exactly what's going on. Again, remember, compare and contrast means think of all the ways that you can express that this process of transcription is similar to the process of DNA replication, and list all the ways in which it's different. All right, so let's start here. So first, here's a comparison. They're the same in that you have to denature the DNA and separate the two strands. Now, in this particular case, we have a template strand and the strand that they're calling the complementary strand. I want you to cross this out in your notes. I don't want you to use the word complementary here. I want you to use the word sense. This is the sense strand. It's exactly the sense strand that we've been talking about before. This initiation site, scratch that out and write TATA box. Now again, remember though, not every gene has a TATA box as its initiation site. Just about half of them do. The other half have other types of possible sequences. But we've seen the gene with the TATA box there. Now remember, how did this happen in DNA replication? What exactly had to occur in order to get this DNA to unwind like this? If you recall, there was an enzyme called helicase that actually did this and separated these two DNA strands. The exact same enzyme is going to operate here. So here's another comparison between this and DNA replication. But it's different. It's a little bit more complicated. What happens first to initiate transcription is this. A particular protein that comes from a different gene, which is called a transcription factor, has to bind to the TATA box, and it's going to bind to the TATA box on the complementary strand, on the template strand, not the sense strand. Okay, now this picture then is a little bit wrong. There's a couple mistakes in it, and I'm going to try to correct those for you. But you notice here, this is the beta globin gene right here. This is the initiation point for transcription, and here is what they're trying to show as the TATA box. Now remember, there's two mistakes here. This should be CATAAA. There's the two mistakes. There should not be a T here. You've seen it. You actually saw the DNA sequence, and you saw it on the definitive website. So this is incorrect. This should be a CATAAA. But at any rate, this is still going to be the attraction point on the DNA for this molecule. It's called a transcription factor. The transcription factor is what gets everything started. What the transcription factor does is it comes and it binds to the TATA box, but it binds to the TATA box on the template, not the sense strand. Right, so when it binds there, it then will attract a bunch of other proteins, and those other proteins will come and bind to this to form a gigantic transcription complex. And that's our goal here. Now, I'm not going to go through all the proteins that bind, but a bunch of them come in, and there's two key ones that I do want to mention. There's this one that's attracted to the transcription factor, which is an RNA polymerase II. RNA polymerase II binds to this, and notice it binds in front of the transcription factor, in front meaning towards the 5' end. It binds to the front side of it, and it starts right there. You see where that green section is? That's where the actual machinery is, where it's going to start making the copy. So the TATA box is back here, but the copy begins here. Now go back to the beta globin gene that I showed you, and you'll see the TATA box is to the 5' end of where the actual transcription begins. And then it attracts a bunch of other enzymes, including this enzyme here, which is a helicase. So the helicase is now part of this transcription complex. There's all a bunch of other things in here which you'll learn when you get your upper division courses. And now we've initiated this transcription by putting in the transcription factor and all the other proteins that are going to now start this process of copying the DNA into messenger RNA. And of course, like I've been saying all semester, this is just scratching the surface. There's an enormous complexity here that I'm just entirely glossing over. I do want to add a couple more complexities though to it, because these are things you're going to see in your upper division cell biology class that in fact you're going to run into it in a lot of different ways. This is one of the key steps in regulation of how many copies of a protein are built in a cell. The transcription factor is one of the key parts of that regulation sequence. Because remember, the transcription factor is what assembles all the rest of that replication machinery. So the cell will control how much of the protein is made by controlling how easily this transcription factor is able to bind to the DNA. There's a couple of different ways it can do it. It can use regulator proteins, which will bind upstream in the promoter. So this is another portion of the promoter. Remember I said the promoter was something that allows the cell to determine when the gene should be turned on and turned off. It does actually more than that. It tells the cell how much of the protein should be made. So the way it does that is this. It attracts other regulator proteins, which again are made by other genes. And those regulator proteins can be of two different major types. One of them is inhibitory. And if an inhibitory protein binds to the promoter, it tends to block or push away the transcription factor, and so it won't be transcribed as often, and therefore you won't get as many copies of the protein made. The other possibility is that it can be an attractive one. If this regulator protein is an attractive protein, it'll attract the transcription factor, allow it to bind more readily, and then that will make more of the protein. Right? So if you need an abundant supply of that protein in the cell, then this is going to be an activator protein. If you want to only have a small amount of protein in that cell, this will be an inhibitory protein, and it's even more complicated than that. It's a bunch of these proteins come together, and their sum total of the interactions determines how readily that transcription factor binds. But there's another thing that's very important. In fact, we're discovering just in the last 5 to 10 years that this is insanely important in determining how much of a protein is made in a cell, and that is other sections of the DNA which are not part of the gene that I showed you. The gene that I showed you is how your book defines genes. That's changing, so you're going to have to keep your mind open about that. Our concept of a gene is changing to the point where we're understanding the regulator sequences that are spread all over the chromosome and in fact spread all over the entire genome for this one gene. These little regulator sequences are called enhancers. The enhancers are something we've known quite a lot about, but it's only been in the last 5 to 10 years that we've started to understand how important they really are. It turns out that essentially every gene has enhancers scattered all over the place. It could be on the chromosome that the gene is on itself, or it could be on other chromosomes. But what these enhancers do is they attract these other proteins called activators. Now notice these are not regulators. They're activator proteins, and they are this region called an enhancer. Remember, the word to enhance means to make better or bigger. That's what this is going to do. This activator protein is going to activate this complex. Now notice it's way far away. It can be thousands of base pairs away, hundreds of thousands of base pairs away, on different chromosomes even. And somehow it interacts with this transcription factor complex, this replication complex, to increase the rate at which you produce this protein, the rate at which you produce the messenger RNA. It does that by doing this. The activator protein is attracted to this sequence of DNA in the enhancer. And then what happens is the chromosome or the other chromosome will fold over on itself, or the other chromosome will come in and allow the activator protein to bind to this replication complex. And when it does that, it releases the replication complex and allows it to do its job and to make the copy of the messenger RNA. So the activator protein, the amount of activator protein and the amount of enhancers for a particular gene will determine how readily this transcription complex gets started, and therefore that will determine how much of the protein is made. Now again, I can't warn you enough. I'm just scratching the surface. There is a huge field of gene regulation and gene expression studies that are going on today to try to get all of the details down, and we know a lot more than I'm telling you. But this is just the beginning, so that when you see this again in your upper division courses, you will at least have heard the terms and know essentially what it is that's going on. All right, so that's how the whole process gets started. You have the transcription factor binds, tracks all the proteins. Regulator proteins will determine how readily the transcription factor binds. Once the transcription complex is made, then the activator protein comes, activates it, and it takes off and does its job. Okay, so that's all of the initiation steps. Once you've got that started, then the RNA polymerase II is going to do its job and start making that messenger RNA. You notice it started there, and what it's going to do then is move down the DNA, and it's going to move only on one strand, not both. The strand it's going to be on is the template, this one here that's labeled template, not this one here that's labeled sense. And what it's going to do is it's going to start making the messenger RNA. And that's what this green object is here. You notice that the green object is lined up directly with the template, and you've got a bunch of these nucleotides that are being put in by this RNA polymerase II. All right, now, I want to walk you through that. So here we are, we've already started the process. We've initiated it. Here's the DNA that's in its native double helix on both sides of this open replication area. Now, what's holding this open? You should know that. I'm going to leave you to answer that question. But the point then is, here, where it's open, is where this messenger RNA is going to be built. All right, so let's pretend that we are looking at the RNA polymerase II, and it's right here, right where the circle is. Now, you can't see it. It's hidden, because I want to show you what it is that it's going to do. Now here we have the messenger RNA. Notice it's running from 5'. It starts on this end and goes this way. Just like all nucleic acids, it's built from 5' to 3'. And you notice that this strand that's complementary to it starts with the 3' end here. So again, it's anti-parallel, just like we've seen a bunch of times so far. Now what's going to happen is this. The RNA polymerase is going to read this template strand, just like the DNA polymerase did, if you remember that. It reads this C, and it sees a C there, a cytosine, and floating in the nucleus are a bunch of RNA nucleotides, which can bind which may or may not bind to that C. So what the RNA polymerase has to do is find the correct complementary one, and of course the correct complement to this is G. So what it does is it puts a G in there, forms the hydrogen bonds here that are complementary to those two. If you remember, there's 3 hydrogen bonds between G and T, and then it forms this bond. So it catalyzes the formation of 2 types of bonds. First, the hydrogen bonds are connected here, and then it makes this connection between this nucleotide and this nucleotide. Okay, we've seen this a bunch of times before. You should remember or recognize that this bond right here is going to be phosphodiester. It's going to go sugar, phosphate, sugar. All right, that's it. That's all the RNA polymerase does, is it lines those things up, finds the complement, and then catalyzes the formation of those bonds. So here it reads the template now, moves down exactly 1 nucleotide, sees a C, grabs a G, and puts it in there. And then it goes down here, sees a G, grabs a C now, and puts it in. Now the next nucleotide is an A. So what should it be? Well, again, we're making RNA, not DNA, so it's not going to be thymine, it's going to be uracil. So there's uracil. Next A, also uracil. T, well, this is DNA, we're going to RNA. The RNA still will be an A associated with that T, so that's an A. And then G binds with C, A binds with U again, and C binds with G. All right, that's all the RNA polymerase does. It just simply walks its way down that template strand, lines things up, and remember the types of bonds that it makes are going to be the hydrogen bonds, which are going to be 3 hydrogen bonds between C and G, and 2 between A and U. And then it catalyzes the formation of these phosphodiester bonds here. All right, so the question is, what end is this? Again, you should know this. The antiparallelism requires that we're building from 5'. This, therefore, must be the 3' end. That's it. That's all that the RNA polymerase II does, is to build that messenger RNA. Now, notice what happens, though. Once we're past this area where the replication fork, or the replication complex has moved, notice the DNA goes right back into its native double helix. Why does it do that? Well, we haven't really talked about the physics of that yet, but it comes down to the fact that once that helicase is gone, and all those structures that are holding the DNA apart have moved on, the DNA up here, this strand, will start to compete with the messenger RNA for the template strand. Now notice they're the exact same sequence. You see that? This is the sense strand, G, G, C, T is replaced by U, T is replaced by U, A, C, T is replaced by UG. So the messenger RNA is exactly the same as this upper strand, but notice the upper strand has nothing to do with its formation. The formation is only made by the template. But the point is that this is complementary to the template, just like the messenger RNA is complementary to the template. So they start to compete for the template, to bind to the template. Eventually, the complementary DNA strand will win because it's at a lower energy state. And we'll talk about this when we get to the thermodynamics section in the next week, actually, as a matter of fact, that once you have the DNA and DNA together, it's at a lower energy state. It's more stable than when you have DNA and RNA together. So once that happens, the RNA has no place to go except off. It just gets kicked off. No special enzymes are needed, nothing special to do that. I mean, there are some things that do this and help. But the point is that this will happen sort of naturally on its own, and no metabolic energy needs to be expended in order to get that to occur. Also notice this. Does this introduce mutations into the DNA? The answer is no, it doesn't. Because remember, what mutations are changes in the DNA sequence. So this doesn't do anything to the DNA. The only thing that is being catalyzed is the formation of RNA. The DNA is just simply there in order to be read or just to be moved out of the way. So this doesn't really introduce much in the way of mutations to the DNA strand. So you might be wondering, well, wait a minute, what if there's a mistake? Does the RNA polymerase ever make a mistake? Like, for example, does it ever, instead of putting in a C here where the G is, it puts in an A instead? And the answer is yes, it can do that. That does happen. But remember, the rate at which that happens is really low. It's on the order of 1 in 10,000. Most of these genes are less than 10,000 nucleotides long. So it's rare. It does happen, but it's rare. But it's also temporary because the messenger RNA is not permanent. If the messenger RNA were permanent, then it'd be a problem that's always there in the cell. But these messenger RNAs last anywhere between minutes to hours to maybe a day. So they don't last long enough that any mistake is going to cause a problem, and even then the mistakes are rare. So they're not mutations. Mutations are permanent changes in the DNA, not changes in the messenger RNA. All right, so that's the elongation phase of transcription. And so we've made now our transcript, and this is what we call the primary transcript. Here's the DNA, the native DNA. It's gone back to its native double helix, and here's the RNA that's fallen off, and then it continues on until we get to this site here, which is the termination site. It ends, the whole process ends in a fairly complex kind of procedure, which I'm not going to spend a lot of time talking about because, again, we're just scratching the surface. But if you recall, there was that sequence at the end of the gene that went AAT and then a bunch of A's, and then 3 TTA and a bunch of T's. That sequence is the end sequence. That's where this messenger RNA transcript tends to be cut off. And what happens is this. Once the transcription complex gets to that sequence of A's, we call that a polyadenylate sequence. Once it gets to that sequence of A's, an enzyme comes and cuts the messenger RNA away, and the transcription continues, and it just continues on until eventually it stops somewhere downstream. But the messenger RNA will stop at that sequence of A's. And then it gets a little bit more processed by mechanisms which we'll talk about in the next lecture, but that's what happens to make this what we call the primary transcript. So like we saw with DNA replication, what I've shown you so far is a highly, highly stylized schematic cartoon of what's actually going on so we can see the events as they unfold. What I want to do is take you to the DNA Learning Center at Cold Spring Harbor and show you an animation that is, again, based at least in part on mathematical models and very detailed research that's done into the shapes of these proteins and how they actually interact, that will give you a more realistic picture of how, in fact, this whole process actually works. So what I want to do then is to get this started, and you can see here the transcription factor already bound to the DNA. This is one part of the transcription factor. And what it's going to do is it's going to attract a series of proteins which will form the transcription initiation complex. And it's moving around here because there's thermal energy in the system. Things are bouncing around because of the energy that the molecules have. And it is still nonetheless able to attract proteins because it has negative and positive charges that, like magnets, will attract proteins like this. Now, what it's attracted here is a complex that includes this blue molecule. And that blue molecule is a combination of the helicase plus the RNA polymerase II. Once it's made this complex, this is the transcription initiation complex, which I showed you before. It has the transcription factor, all those other proteins, plus the DNA polymerase II. Now it's bound to the DNA and it's stuck. It won't start until it's given an enhancement from this activator protein. The activator protein right here is bound to the enhancer. Notice how the chromosome folds over. So this is not really even part of what we traditionally call a gene. But the moment this binds, watch what happens. This will come in here, bind to a specific spot on here, and then boom, this thing takes right off. That's about right. And in fact, this is approximately the right speed, I believe, in terms of how this messenger RNA is being made. But what you can see here is the RNA polymerase III right here, this blue complex here, making this messenger RNA from this DNA strand right here. And again, this is about the right speed. So it cruises along here and it makes this, this messenger RNA at about this rate. And you can see all these yellow nucleotides being pulled into one particular spot. And again, when you get your upper division courses, you'll study the whole physics of all of this and how this works. But this gets pulled in by, again, those positive negative charges that act like magnets and pull those nucleotides in the correct orientation so that they can be made. And then that's showing a very stylized sort of ending of the process. That's not quite how it really ends, but that is a very, very quick and pretty accurate view of transcription.